Okay, good morning and hello to those of you who've joined in. I dare say we'll probably have more people joining in as the class goes along. Again, just please, when you join, please mute your microphones, and but you are welcome to interrupt anytime, go off mute and pose questions anytime that you want to. So this morning I'm going to finish discussing our section on the protists, and this last section about the protists, it segues really nicely into our succeeding sessions on animal diversity and then on the other groups of animals that we're going to talk about, as you'll see. So our very last group of protists that we're going to talk about, I know that these groups seem complex and the names are difficult to remember and everything, but always go back to that very first diagram, the first phylogenetic tree that showed all 5 of the groups that we have talked about. And this is our very last group and a really important group for many reasons that I'll point out. This is the group called the Uniconta. And it's for us particularly significant because it includes us, the animals. And the Uniconta is a very mixed group, as you'll see if you have a look here, that all of those which are highlighted in this yellow beige color, these are all protists, but the others here, the fungi and the animals, are obviously not protists. They are, for the most part, except for some fungi, mostly multicellular. And they have completely different characteristics that set them apart from the protists. So what are they doing here? Why are they placed here in the Uniconta, together with all of these things which are clearly protists? Well, that's an interesting story. And as we go along, I'll point out several things which link these groups all together, but which also tell us a story about how multicellularity evolved, and multicellularity not only in the fungi, but most importantly in the animals, which we're going to spend some time talking about the animals in general. When we do, always remember that the origins of the animals lie here. They lie in the protists, and that is reflected in the fact that the animals are included in this group here, the Uniconta. Okay, so let's have a look at obviously there's a huge amount of diversity in this group, the Uniconta, but there are things which unite them as well. So let's have a look through. First of all, there are 2 clades within the Uniconta. The first are called the amoebozoans, and the other is called the Opisthokonts. And the Opisthokonts includes the animals, together with some protists as well that we'll talk about. The amoebozoa, well, the name tells you. It tells you one of the most important characteristics of this particular clade, and that is that most of them have, at some stage in their life cycle anyway, they have an amoeboid form, a single cell which is motile by producing pseudopodia, which enable it to move and with which it feeds. That they are able to take in material from the outside by forming a vacuole. They put out pseudopodia, which envelop whatever it is that they want to eat, and they ingest it in a food vacuole. So that we'll see in all of these groups: the slime molds, Gymnamoebas, and entamoebas. And we're going to spend some time talking about the slime molds in particular, because when we look at the slime molds, we see something very, very interesting, and that is the beginnings of multicellularity. These others are solitary individual amoebas, but something fascinating happens in the slime molds. So the amoebozoa, they have amoeba with pseudopodia, There's three groups: slime molds, gymnamoebas, and entamoebas. Entamoebas I'll only just mention as we're in passing. So there are two groups of slime molds that behave really quite similarly. It's that there is a distinction in their morphology that separates them from one another. The one group are called the plasmodial slime molds. Don't confuse that with the organism that causes malaria, which is called Plasmodium. The plasmodial slime molds are a little bit different, and that is at one point in their life cycle, individual amoebae get together and they actually cluster together and they lose their cell membranes and they become a single mass, which is multinucleate. So it's not multicellular, but it is multinucleate. And there are some other distinctions, but that can behave as a single organism. So it's many amoebae get together and they form something that behaves like a single organism. Now that in itself is interesting. It carries on moving around, feeding, etc., but can then go on to form a feeding body. So that's all I want to tell you about the plasmodial slime molds, because we want to actually talk more about this group called the cellular slime molds. And the most famous one of all, if you were in lab, if you were doing a lab, you would probably see this in lab. This is an organism called Dictyostelium. And it's really a very fascinating organism. And when we study Dictyostelium, we see the true origins of multicellularity. That is what we believe. I'll show you now. Here's the life cycle. It looks pretty complicated, but it's actually not that bad. For the vast majority of the amoeba's life, they are haploid, N. And only under certain environmental conditions, they will get together to actually fertilize one another. Now, for the most of the life, they wander around as individual amoebae. They occur in damp environments, very common in rotting logs and in understory of forests, things like that, and very easy to recognize. Excuse me. And that is because they are often very brightly colored. And you turn over a log or you look on top of a damp log, sometimes you'll see these brilliant scarlet, yellow, or orange masses. Those are slime molds. For the most part, they wander around as individual amoebae, but they can respond to one another in various ways. And the first is, under certain environmental conditions, they will get together and fertilize. In these individual amoebae, they're not male and female really. They're often designated as positive and negative strain. And you have to have a positive strain and a negative strain meet up and unite, and they very briefly form a zygote, which is 2N, and that's the only time that they are diploid. That zygote then undergoes rapid division and forms more amoebae. So you can end up forming millions upon millions of amoebae by sexual reproduction. These wander around individually until certain environmental cues get them to release signals, which the other amoebae pick up. And they begin to congregate. They aggregate together and they stick together. They may, in fact, form a covering around the outside. It forms a visible little slug, which is multicellular. In this one, unlike the others, the plasmodial ones, in these ones, the cell membrane is preserved. So they are truly multicellular. And the amazing thing is They behave as a single organism. Here's one here, like a little slug-like giant amoeba. And that amoeba can wander around. It can still feed and everything else, but they begin to send out signals, and these amoebae begin to aggregate together, and they start to form a really large mass, they interact with one another, and they begin to develop. And what they form are these fruiting bodies here. These are elaborate. These are elaborate structures. This means that these cells have to be communicating with one another very closely. There is all sorts of genetic information being switched on and off in this mass to produce these fruiting bodies. I can't overemphasize the significance of this, because this means that this slug is now behaving as if it was a multicellular organism, and it is specializing parts of itself for reproduction. So that is why we're so fascinated by these cellular slime molds, because we believe that in this whole process here lies the origin of multicellularity in animals, and that it's through these kind of processes that multicellularity evolved. In actual fact, I won't belabor the point, but if you look at the molecular biology of cellular slime molds, and you see which genes are being switched on and off when they're differentiating like this, and you look at the proteins and everything that tie these cells together, we see the same genes being switched on and off in the animals, in animal cells, and we see the same proteins being used to bind animal cells to one another to form the organism we call an animal. So there are very strong links between these cellular slime molds and what is happening in animals. They are not ancestral to the animals. It's just that it tells us something about the kinds of processes that are possible in the Uniconta, which could have led to multicellularity. So here's the next group in the Uniconta, and this is probably more familiar to you. And this is the common amoeba. There are many, many, many different amoeba in freshwater, soil, in marine systems, and the amoeba, characteristically, they are individual single cells like this. This one is moving in this direction here, and this here is the pseudopodium being pushed out. Pseudopodia means false foot, and here it is being pushed out in front so that the cell locomotes. It has a cell membrane. There's no cell wall around it at all. These usually have a single nucleus, and they can get quite large. These amoeba can be just large enough that you can see them with the naked eye. Extremely, extremely common. So they feed, you can imagine if there was something that they could feed on here, they would wrap these pseudopodia around it and ingest it in a food vacuole. The freshwater amoeba, just like those freshwater ciliates that I told you about, they have a problem that the water continually pours into the cell due to osmosis. And as a result, usually somewhere inside them, I can't quite see one on this photograph, but you will see very often a large, clear vacuole which pumps water out to the outside. It pumps water out as water floods across here. And that's the contractile vacuole. Amoeba usually only have one. The ciliates have two. So these are the amoebozoa. I'll also just in passing mention, sorry. Oh, wait, yeah, all right. The amoeba are predominantly heterotrophic, but I will mention one thing, and that is that there are amoebae which have which keep symbiotic algae or cyanobacteria in their cytoplasm. So that's quite interesting because it begins to sound like they are actually on their way to becoming photosynthetic, but those the organisms that they keep inside to photosynthesize are actually relatively independent. You can culture them outside of the amoeba. So it's a symbiosis. It's not true endosymbiosis. So let's just go back here because I just want to make it clear where we are. Okay, I'll just, in passing, I'll just mention this. This little group here, we've talked about slime molds, Gymnamoeba, ordinary amoebas. This group here, the entamoebas, very, very similar to the Gymnamoebas. They're much smaller usually than the Gymnamoebas, but the thing that characterizes them is that they are parasitic. So, for example, there is a very famous one in human beings called Entamoeba histolytica. And that is the organism which causes amoebic dysentery, very important disease worldwide. It is transmitted in contaminated water and food that has been contaminated with feces. So in areas with poor sewage, poor infrastructure, amoebic dysentery is extremely common and very debilitating. And in fact, it can be fatal. One of the things that entamoeba can do is that it can pass into your bloodstream. It damages the walls of the gut so badly sometimes that it can pass into the bloodstream and it ends up lodging particularly in the liver and forming abscesses in the liver. In most cases, amoebic dysentery passes off. People are sick for a while. And then it passes off. In other cases, it can be a chronic illness, which can be very debilitating. So those are the amoebozoa. And remember them, they're a really important group to remember. The next group that we're going to talk about are the Opisthokonts. And this is a clade. Here you can see they have a common ancestor, just as the amoebozoa here had a common ancestor. And the Opisthokonts of this group here are extremely diverse, and they include single-celled organisms, here, the nuclearids and the choanoflagellates. But we also see in these two protists, in this protist group, particular, we see again the possible origins of various things which characterize animals. Strange though it may seem when you see what I'm talking about. We're not going to talk about the nuclearids. We're not going to talk about the fungi except just to refer to them as I have done. We're not going to, at this point, discuss the details of the animals because we're going to start talking about animal diversity in the succeeding section. But we are going to talk about this group here, the choanoflagellates, because when you see the choanoflagellates, when you see a choanoflagellate, greet it nicely, be polite, because you're greeting your great grandparent. Okay, here in this group here lie, we believe, all the evidence tells us that it does, here lies the origin of the animals. The ultimate ancestor of the animals, we believe, was a choanoflagellate in this group, the Uniconta. And they obviously, the group includes things which look wildly different to one another. Hold on, let me just flip through. Here we are here. So this is a choanoflagellate. That means a collared. Choana means collared. It has a collar. And look carefully, and you'll see. Here's the cell body. Here's the cell body here. There. And here is the collar. And the collar consists of columns of protein, which are united together to form a net. And inside here, there is a flagellum, which beats. And it pulls water through here and pumps it out of the collar. And this is the way in which this little cell feeds. It is a filter feeder. The particles are ingested. Particles trapped here are ingested by the cell. So we look at that and we say, what on earth, how does this tell us anything about the origin of the animals? Well, the important thing about the choanoflagellates is that they can get together. There are many which are solitary individual cells, but there are others which cluster together. Interestingly enough, when we look at how they cluster and we look at the proteins involved in binding them together, we find that they're the same kinds of proteins that are present in the cellular slime molds, and they're the same as the proteins that animals use to bind their cells together. So second of all, when we look at the molecular biology, when we look at the DNA, we see that there is clear evidence that that something like a choanoflagellate ancestor was ancestral to the animals. The key, we believe, is when we look at the strange group of animals called the sponges. Now, the sponges, we regard them as animals, although some molecular biologists would actually like to have them classified as protists, as colonial protists. So when I tell you that, you realize they're sitting in this strange position. We can't quite tell, well, what is an animal? An animal is a multicellular heterotroph. That's almost the simplest and most direct definition, the multicellular heterotroph. Well, the argument with sponges is, are they multicellular or are they just colonial? The choanoflagellates like this, we regard them actually as being colonial, not multicellular, because the cells don't really interact with one another that much. They just sit together. But in the sponges, there are many, many different kinds of cells. The important thing in sponges, they don't form tissues, and you'll see why that's important in a minute. But they do interact with one another. They do interact. There are feeding cells, for example. And when we look at the feeding cells in the sponges, what do we see? We see in sponges, they're called choanocytes, collared cells, but they are identical to choanoflagellates. It looks very much as if these colonies of choanoflagellates in evolutionary history got more and more and more elaborate and the cells interacted more and more and more with one another, and they formed these large collections which went on to evolve to be the sponges. So we're really interested in the sponges. They're a strange group in relation to the animals, but you'll see it when we talk about animal diversity, that they are important in our ideas about evolution of the animals. Okay, so now we're going to move on to our next section, and this is to provide an overview of animal diversity, because we are going to be talking about animal diversity in some considerable detail. So leaving the protists behind, let's talk about the animal kingdom and realize we sit, of course, right in the middle of the animal kingdom. We are clearly animals, and we share animal characteristics with many, many other multicellular heterotrophs, which we would classify as being animal. There are about, let's say, currently we identify 1.3 million living species of animals. It's probably the number of living species of animals is far greater than that, where there are many, many, many which we know nothing about, which have never been identified, particularly in the things in groups like the insects. Every day, new species of animal are identified. Animal diversity is absolutely enormous. And there are many different animal groups, which honestly, when you look at them, you wonder how can we say that they're even related, because they are so very different to one another. All of the groups shown here in this beautiful picture, these are all animals, including the divers, all the way down to things like sea anemones, corals, that sort of thing. And we're going to hear about all of them and about some ideas about how they are related to one another. So here's the definition. Animals are multicellular. They're heterotrophic eukaryotes. But here's a characteristic that you may not have heard of. And that is that the animals characteristically have their cells organized into tissues. That is, they have groups of cells which are similar very often in structure and which are grouped together to form a single function. And very often those, in most cases, a tissue is separated from other tissues by some sort of membrane, by some membranous structure. This won't mean much to you at the moment, but it will in a while. And that is to say that those tissues, the tissues which make up the animal, develop from embryonic layers. And those embryonic layers, where they come from, is very significant when we look at how the different animal groups relate to one another. So we spend a lot of time when we talk about the evolution and the phylogenetics of the animals, we actually look very carefully at embryonic development as well. So animals are strictly heterotrophic. Some animals may have associated symbionts, but animals are fundamentally heterotrophic. So most animals have to eat their food. That we'll hear was a significant development in animal evolution. So they eat either living or non-living material and largely depend on other organisms which have already either photosynthesized or have eaten things which have photosynthesized. They are depending upon already fixed molecules, already created molecules, for both their carbon and for their energy. Animals are distinguished from fungi, which are the other major group that is largely multicellular, because fungi do not ingest their food. Fungi release enzymes into their environment, digest molecules in the environment, and then absorb the simpler resulting molecules through their cell membrane. Instead, animals need to actually ingest food, and the cells of the animal, digestive cells of the animal, need to take in food, and then it is distributed to the rest of the animal. So there are some, first of all, some of the characteristics that I've given you, apart from being heterotrophic, some of the characteristics, multicellularity, this sort of thing, also apply to plants. But plant cells and animal cells are quite different. First of all, obviously, animals are heterotrophic, plants are autotrophic. But plant cells are distinguished from animal cells because they have a cell wall. Animal cells have no cell wall at all. They only have the cell membrane around the outside of the cell. Because they lack a cell wall, which is what binds plant cells together and forms the body of the plant, animals have to have some other mechanism to bind their cells together. And they largely depend on a protein called collagen. There are different forms of collagen, but you can just use the blanket term collagen. And this is a structural protein which ties cells together. And that is important because the tissue, because animals very often have organs which consist of many different tissues which are bound together to form a single unit which performs distinct function. Organs may do more than one function, but they have distinct functions. And those functions are the result of certain tissues with certain functions being tied together. There are some tissues which are really characteristic of animals, which no other living organism has. And these are particular nervous tissue, that's one thing, and muscle tissue. These are unique to the animals. So the animals also reproduce sexually. They have a diploid stage which dominates the whole life cycle. Most animals, there is only a very brief haploid stage, and that is in the production of the gametes. When animals reproduce, they produce a zygote, which is formed from union of a sperm and an egg, usually, excepting in the most primitive, some exceptions. The zygote undergoes division, and those divisions are called cleavages. And the cleavages lead to the formation of, very rapidly, lead to the formation of a multicellular ball, which is hollow on the inside, and that is called a blastula. That blastula then undergoes a process, an elaborate process called gastrulation. And gastrulation converts this single cell thickness wall of the blastula. It converts that into multiple layers. And those multiple layers go on to form all of the tissues of the body. That process of converting that one layer into multiple layers is called gastrulation. And biologists always say there are three critical events in the life of an animal. And those are fertilization, and then gastrulation, and then puberty. So those are the three. And of all of them, gastrulation is the most critical. It is absolutely, gastrulation is the one process which really can go wrong, can go badly wrong. It is extremely complex. And many, many embryos which undergo gastrulation fail and are lost. In human beings, we know that this happens frequently, and somebody who who's pregnant with an early stage embryo like that won't even notice that it has happened. It is just lost. But gastrulation is a critical point in an animal's development. I can't show you this little movie. It doesn't work on Zoom. But please just go here to this YouTube video about sea urchin development, and just see it takes you up to through the early stages of fertilization and then formation of blastula. And I think it stops just before gastrulation. So here's what the process of gastrulation, of fertilization and then gastrulation. And this here would be for something like sea urchin. And here's a zygote. The egg has been fertilized by a sperm and it is diploid. It undergoes mitosis very rapidly in order to form this little ball of cells here. And those carry on dividing to form this blastula. And here it is here. This is the blastula here. This hollow in the middle is called the blastocele. So blaster with this layer here, single cells. And this is the process of gastrulation. There are various ways in which gastrulation can occur. It's easy to understand in the sea urchin because what happens is it invaginates like this. So you can see this begins now. This here is moving in to form a second layer on the inside, and that is the process of gastrulation. The opening of this tube that is formed here is called the blastopore. And I'll just warn you now, keep your eyes on this, get this kind of picture into your head, because the fate of the blastopore is really important. In phylogeny and in evolutionary history, what happens to the blastopore is a defining feature of different groups of animals. This hollow here, note that it's not the same as the blastocele. Here's the remains of the blastocele here, there. This is called the archenteron. I'm not going to spend a lot of time talking about embryology, but try and remember these different terms. It'll help you to actually follow what is happening a little bit later on. So that is the process of gastrulation. Now, I'll just tell you right now that in some animals, they only have two layers, this one on the outside and this one on the inside. So they only have two. Their gastrulation would stop there. And we will see a group like that. Other groups, however, end up with a third layer on the inside here. And that can form by various means. There are two principal ways, which I'll show you, but just get this picture in your mind. This is the early stages of gastrulation here. Here's gastrulation occurred there. We have a space there, and we have two tissue layers, one on the outside, one on the inside. In some, as I've told you, you may go on to have a third layer laid down on the inside. So a lot of animals have a stage where the embryo initially forms a larva, a juvenile form, which is not sexually mature and often very, very different to the adult. That larva will then go on to, at some point in its development, cues are switched on and it will undergo metamorphosis. It will transform into an adult. That we'll hear, as we talk about the animal kingdom, we'll hear about these different larval forms and the different ways in which they actually can end up metamorphosing into an adult. Now, what a larva is distinguished by the fact that it is morphologically distinct from the adult. It looks completely different. And the simple example to give you is something like a butterfly. Okay, a butterfly, the larval form of a butterfly is a caterpillar, looks completely and utterly different and behaves completely differently to the adult. That larva, in the case of a caterpillar, it undergoes metamorphosis by forming a pupa. And then the adult hatches from that pupa, looking completely different and behaving completely differently. They're like two different animals that are linked by their life cycle. A juvenile is different. A juvenile resembles an adult. Now that's like us, or like fish, or anything like that. A juvenile is incapable of reproduction, but essentially looks like the adult, and in most ways behaves like the adult. It's just that it's not sexually mature. So this is a distinction that should be clear in your head. Okay, let's have a look at some of the ideas that we have about the history of animals, about how they evolved. Well, in terms of the whole geological history of the Earth, which is more than 4 billion years, animals are a relatively recent arrival because they have been present on the planet for just over 500 million years, about 550 to 600 million years. The animals have existed. In order to look at the origins of the animals, we have to look into the fossil record. And when we look in the fossil record, we realize that the vast majority of kinds of animal that have ever existed have actually gone extinct. We believe that all animals share a common ancestor, a single common ancestor. And our current thinking is that ancestor, as we've already discussed, is something like this organism here, which was a choanoflagellate. So here's a choanoflagellate. Here's a living choanoflagellate. This is probably marine, and you can clearly see the same features. It has a single nucleus. Here's a cell body there. You can just see the collar there, and here's the flagellum there. But here is a colonial choanoflagellate. They are joined together. The cells do not really interact very much, although they can direct their movement. These flagella not only conduct water into the collar, they also allow the organism to locomote. And when they get together like this, they can interact insofar as they can direct their movement to stay in optimum place. So this is what I meant when I said greet your however many hundreds of thousands great-grandparent. Here it is here, looking at you. And this is what we believe happened. I've hinted at this already. We believe that, in fact, the choanoflagellates were in some way ancestral. Something like a choanoflagellate, rather, was ancestral to our modern choanoflagellates and to this group called the sponges. And that the sponges and the animals also share a common ancestor. So we have to say that the origins of the animals remain obscure. We don't definitively know which was the answer, but this is our current idea of it. There was something like a choanoflagellate that became colonial. The colonialism of the organism led to multicellularity. And this is all that we can say about the sponges. The sponges are multicellular. They are definitely multicellular, but they do not have the one characteristic which defines the animal. They do not have tissues. There are no tissues present in the sponges. They are definitely multicellular. The cells of the sponge interact with one another. We have feeding cells which feed the rest of the sponge. That's number one. There are other cells which behave something like muscle cells. They're individual cells, but they link up with one another and they are able to contract the whole cell. Each individual cell doesn't work exactly like a muscle. The cells bunch up like this. And there are not all sponges, but some sponges, if you poke them, you can see them visibly contract like this because of so there are cell interactions, but they are not forming tissues, which is characteristic of animals. So the sponges sit outside. They're a group of which cause great controversy among scientists. There are many scientists who would rather have the sponges removed and put into the protists, but most people regard them as being linked to the animals, but not really true animals. All of the other animals, no matter how diverse they are linked by those characteristics such as the fact that they have tissues, that they're heterotrophic and have tissues, which are distinct, and they have nervous tissue, and they have muscle. So the origins of the animals lie very far back, and go back to our previous class where we had the whole geological time laid out as a chart for you, and have a look there. And you'll see that the very first animal groups occur here in the fossil record. They may have occurred before, but we see them in the fossil record about 565 million years ago. I believe there is one which has now been found as far back as 600 million years ago. The early animals here in the Neoproterozoic was a distinct group of animals called the Ediacarans. And here they are here. Very strange. We do not know how they functioned. We know that most of the Ediacaran fossils looked like flat and leaf-like kind of animals. We surmise that here are some reconstructions here. We surmise that a lot of them were floating forms, and that some were embedded in sediments. One of the things that is missing from any of the Ediacaran fossils, none of them seem to have a gut. None of them seem to have a mouth. So that is mysterious. What are they actually feeding on? They are heterotrophs. So how are they surviving? Well, many people think that most of them had symbiotic cyanobacteria or algae associated with them, and that's what they depended on, and that they could also absorb molecules from the water around them, molecules resulting from natural decay or whatever. They did form a very distinct fauna in the fossil record, but we have very little idea about what they actually look like or about how they functioned. A mysterious group, successful for a long time, but then they suddenly die out, and they die out absolutely completely. Towards the end of the Ediacaran period, we start to see some groups arising, and you can see some here. The jellyfish here start to appear in the fossil record towards the end of the Ediacaran. Well, what do jellyfish do? How do jellyfish survive? Jellyfish are predatory. They are predatory. They actually grab hold of living organisms, other animals, maybe Ediacarans at this time, pull them in and digest them and eat them. That is a very new development towards the end of the Ediacaran. Predation starts to arise. And we believe that predation actually spurred many of these early groups, probably very obscure groups, but it spurred their evolution. They suddenly began to diversify. Just picture what happens. Imagine here in the Ediacaran, take out the jellyfish. Just imagine the Ediacaran with these floating forms and some of these simple things embedded in the sediment. Everybody's happy. It's all kumbaya because nobody eats anybody else. It's all peaceful and everything. And all of a sudden, something evolves which can eat you. Well, what is going to happen? You've got an enormous population of very diverse organisms floating around, which are now being preyed on. Who is going to survive? Who's going to survive out of those lineages when you've got a predator after you? The ones that survive are going to be those which have an advantage. And single advantage that something might develop is something like an exoskeleton, some protective mechanism. Okay, so now things start to develop protective mechanisms. What happens to the predator populations? What is going to drive evolution of the predator populations? Better predation, things which have better mechanisms for tearing open those exoskeletons, etc., etc. The development of predation drove an explosion of diversity and made these groups, forced these groups into suddenly developing characteristics, elaborating characteristics, which led to a huge boom in diversity. And that it's so rapid in the fossil record towards at the end of the Ediacaran that it marks the beginning of the Cambrian. And the Cambrian era is a time when there was just a huge explosion of new groups, elaborate, highly predatory, some of them, and incredible forms. And here at the end of the Ediacaran and the start of the Cambrian lie the origins of most of the modern groups. We believe that explosion was driven primarily by these predator-prey relationships, but we also notice that there are some environmental changes as well. There was a change for some reason, there was a change in the atmospheric oxygen levels, which may have been due to plants moving onto land, the development of land plants. There is one thing I've never talked about it much. That is, there is also, we believe, the development of a particular gene complex in the genome. And this is called the Hox gene complex. I have not mentioned it before. If you're interested, just go to your textbook and look up Hox gene in the index, and you can read. There's quite a lot about it. It's quite complex, but extremely interesting, because the Hox gene complex is a gene complex that governs body plan. And I'll have more to say about that now. There are distinctive ways in which a body can be constructed, an animal body can be constructed. And that is determined by the Hox gene. And mutations in the Hox gene drive the development of new body plans in the animal kingdom. This is a picture of the Cambrian. And you'll see here some absolutely extraordinary animals. This one we've seen before. This is Hallucigenia here. And exactly what it was, we don't really know. But it appears in the fossil record several times. Spiky. We believe that some of them were locomotory, but all of them probably protective. This is an amazing animal called Anomalocaris. You don't need to remember the name. This is Anomalocaris, a very, very fierce predator that has strangest mouth. Here it is here. These enormous, whatever they were, antennae, or we presume that they're equivalent of antennae, but they were apparently used to grasp prey and pull it into the mouth. And the mouth is unlike animal mouths usually are. It's like a ring structure that can open and close, and it had chitinous teeth that could chew up. These could get very large, several inches large. This here, behind here, is the ancestor of a major marine group that went on to be dominant in marine systems, called the trilobites. And the early trilobites were here in the end of the Ediacaran and in the start of the Cambrian explosion. The trilobites persisted all the way through to a mass extinction that I told you about before, and that was the Permian extinction. The Permian extinction rendered the trilobites extinct. Sponges hanging on from the end of the Ediacaran. Many things which we would actually recognize today. This, don't worry about what it is, but this worm here, it occurs in Cambrian fossil record, but we can find worms just like this in marine systems today. So many of the groups, elaborate structures. Many of them became very successful. Most of them have since gone extinct, but we see in them the roots of modern groups. And the one that we need to look at in particular, we're not going to discuss the details of it. I'm just pointing out to you here, this one here. Believe it or not, we'll hear about organisms which are look very, very similar to this, which are our kissing cousins. This is a primitive chordate. That is our phylum. Our phylum is the phylum Chordata. And here is the ancestor. At the end of the Ediacaran and into the Cambrian, this is an organism which is a chordate and is closely related to us. From something like this, an ancestral chordate like this, developed all of the chordate groups. We have invertebrate cousins, but all of the vertebrates and everything are linked very closely to this organism here. At a very, very interesting time. If you are interested, there is a very good book called Amazing Life by Stephen Jay Gould, and very easy to read, and it's all about the Cambrian explosion. Okay, so I'm going to finish by just mentioning that I told you about the Hox gene. And the Hox gene governs the body plan, and the animals can be characterized and categorized by their body plan, and that is a distinct set of morphological traits which reflect developmental traits. And the easiest way to think about body plan is to think about symmetry. How are organisms symmetrical? And just think about yourself. To finish off with, just think about yourself. How are you symmetrical? You're symmetrical from side to side. Not exactly, but more or less. We are symmetrical from side to side. That we call bilateral symmetry. But think of a jellyfish. How is a jellyfish symmetrical? A jellyfish is symmetrical around. It is radially symmetrical. We'll talk about that more next time. Just to finish off, I just want to let you know, first of all, there is a new assignment which went up on Wednesday. There is really no due date for the assignments. I leave them up for the whole semester. But do them as soon as you can after they appear, because they lead you through material which we've already covered in lecture, and it's a very good way of revising. Then go back to the assignment when you're revising for tests. That's the first thing. Second thing is, please look on the announcements on Canvas, because I have put up an extra credit assignment on radiometric dating. So please refer to that. And then I'll see you again on Monday. Monday is a live class again. But before we go, anybody got any questions?
I have a question. So the assignment on my lab and mastering that says the due date is May 14th. The due date is really not May 14th. We can do it now?
You can, oh no, you can, that's just to keep it open for the semester.
Okay.
That's all. That's all. I don't, the assignments, other professors set like a 1 week or 2 week cutoff, you know. that you have to do it by. But I don't like to do that. I like them to be available to you throughout the semester. But do it as soon as you can.
And will we be able to like redo the assignment if we don't get the grade that we want? Can we do it again, or we can only do it once?
I think I've set it so that you can redo it. I think I'll check, but I think I've set it, I normally do, I set it so that you can redo it. Because the whole point about the assignment, it's not a test, you know. It's for you to actually get the information in your head. So I'll check and I'll change it. If I haven't said it that you can repeat, I will change it so that you can. Anything else?
Okay, thank you.
That's a pleasure. Okay then, everyone, I'll see you on Monday then. And have a good weekend. Stay safe, wear your masks, wash your hands, socially distance. Please be careful, okay?
Okay, enjoy your weekend, Professor.
You too.
Thank you, Professor.
Thank you, Professor.
Bye, bye, bye.